Hello my dear children, Namaste and welcome back to yet another session of our newly launched ICSE series. Now we have already started with the very first chapter of biology of your ICSE syllabus, as you know, and we had an amazing session, right? We had a live session and all of you participated really well. So mitosis is one topic that most of you really wanted a lot of clarification on, right? So as you know, we've already set the context for what mitosis is or what leads to mitosis in our previous session, because we explained what cell division is and all of those things, right? So we will start with a quick recap of that. But before that, remember this homework question that I had given you? Now, I got some wonderful answers from a lot of you. Some of you were very quick to give me the answer right there in the chat box. Some of you put it in the comment section. But there was one answer that I found quite well explained, which was by Siddhi Sharma. Thank you very much, Siddhi, for giving me such a wonderful answer. All of you can have a look. It says red blood cells have a nucleus when they are produced, which is yes, very true. But they lose their nucleus when they are mature, that is, they become, it's in fact enucleated, right? RBCs, as they mature, they lose their nucleus and become enucleated. It is so because the cells become biconcave, thus increasing the surface area for absorbing more oxygen, right? Because RBCs, the basic function of red blood cells is to carry around and transport oxygen to different parts of the body, right? So because that is their major function, they have to be biconcave in shape so that there is more surface area for carrying around more oxygen, right? So in that process, what happens is the space in between is increased, thus more RBCs can be accommodated in the same space. In fact, the truth is that it loses its nucleus and saves up that much of extra space to accommodate more oxygen. So thank you very much, Siddhi Sharma, for giving me this answer. Today, again, at the end of the session, you will have one more question. Let's see who gives the best answer for that. Okay, so let's continue. Let's start with a quick recap of what we have done in the previous class. Cell cycle, we have seen that it includes, it's the series of events that occur between the formation of a cell until cell division, right? Between cell formation and cell division, what all happens? That is exactly what cell cycle is. And broadly, it's divided into two stages, which are interphase and the mitotic phase, right? And in our previous session, we have just explained what interphase is. Remember? Because it's a very important step. And interphase is the stage during which the cells, a typical eukaryotic cell, prepares itself for cell division. And we've also seen that the ultimate goal of a cell, of any newborn cell, is to divide, right? So just to divide, how it prepares itself is what interphase is all about. And we have seen that it's divided further into three stages. G1, or the gap 1 stage, wherein the cell grows in size. The second stage is the S phase, wherein DNA synthesis or replication happens. And the third stage is again more of cell growth in the G2 or the gap 2 phase, wherein the nucleus also becomes bigger and the cell again physically grows larger. And once it has completed G2, the cell is ready to enter the mitotic phase, the first stage of which is karyokinesis, which is nothing but mitosis, the topic that all of you have been waiting for. So let's look at what happens in mitosis. Initially, I don't want to start off with the definition. Let's first understand what happens in mitosis and then come to all other related aspects of it. Okay, so let's get started. Now, I'm sure all of you are aware of what's happening in these two, right? Let's look at the very first scenario. This one, wherein we see different stages in the life of a human being, right? It's starting with a newborn infant, and then a crawler, and then a young kid, and then a boy, and then a man. Now, all of us have been going through this, and we realize how much we have grown physically and how much we have grown up ever since we were born and we came into this world, right? So how do you think this was possible? The very basic thing that has happened is at the cellular level, which is the division of cells. Constant cell division is what has led to physical growth in all of us, right? So the second one, look at this. I'm sure all of us have experienced this at least at some point of life or the other. It's just a wound. We must have fallen down. We must have had minor cuts somewhere on our arms, legs, knees, whatever. It happens to all of us as children, as adults. It keeps happening, very natural, right? So it happens that we have a small cut or a small wound, then we just wash it, we clean it, we apply some medicine, and we cover it with some kind of a plaster, and then we may even forget about it, right? Later on, after many days, when you look back at it, you may realize that it's completely gone and you have a fresh layer of skin there. How does that happen? That is exactly what skin repair is, and that again is happening as a result of cell division. So growth, repair, right? Something else that we are familiar with is the growth of nails. We have to keep trimming our nails, right? Regularly, at least once a week. For some people, it's once in 2 weeks, whatever your growth of nails is like. And again, your hair also keeps growing, right? For some of you, it may be very fast. For some of you, it may take some time. Whatever it is, even nail growth and hair growth are all happening in our bodies as a result of cell division. And what kind of cell division? It is a type of cell division called mitosis. That is exactly what we are going to be discussing today. The stages of mitosis, right? These are broadly the stages of mitosis. It's divided into prophase, metaphase, anaphase, telophase, and finally there is a stage called cytokinesis, which is the final splitting of the cytoplasm or the cell of the parent cell into two daughter cells, right? So in fact, I would like to add interphase here so that the context becomes easier for you to connect, right? Let's add interphase here, because this is what we've done in our previous class, right? So after interphase, a cell would directly enter prophase, and then it goes to metaphase, anaphase, telophase, cytokinesis. And what happens to the two newly formed cells? They would individually go into their respective interphases. So this again continues as a cycle and keeps happening. This is basically the cell cycle. Now remember, in one of my previous sessions, I told you a trick to memorize the order of this, right? For those of you who find it difficult to remember the order for this, there are some tips and tricks, but mnemonics do work well here also. So I remember giving you an example. In my case, I think the easiest for me to remember is I prefer Malayalam and Tamil. I for interphase, P for prophase, M for metaphase, A for anaphase, T for telophase, right? I've not included cytokinesis there because it's actually an additional step which happens after mitosis. So I, P, M, A, T. This is basically the order in which cell cycle happens, and this is followed by cytokinesis, right? I hope that's very clear. Have these names in mind. Now let us start by looking at them one by one, okay? Now, this is what prophase is all about. Initially, we know that the nucleus of a cell has a network of thread-like structures called chromatin, right? It's just loosely lying around there. In prophase, what happens is that the chromosomes condense into X-shaped structures. What are those X-shaped structures? The structure that we know about, the structure of a chromosome that we are all familiar with, right? Something like this, the X-shaped structures. Okay? This is roughly what happens in prophase. Chromosomes end up condensing into these X-shaped structures, which can easily be seen under the microscope. Now, this image that I have at the top is actually the view that you can see under a microscope, and this is just a drawn image to represent this, okay? So you see that the chromosomes are arranged as an X-shaped, and we will talk about the remaining structures as we go along, okay? So these blue X-shaped structures are our chromosomes. Okay. Secondly, at the end of prophase, the nuclear membrane becomes less prominent, releasing the chromosomes. Now we know that the nuclear membrane is the layer that surrounds the nucleus, the layer that protects the nucleus, basically the outer covering of the nucleus, right? But you can see here that the nuclear membrane is drawn as dotted structures, right? Do you see that? This is what the nuclear membrane is. Okay? So it becomes less prominent towards the end of prophase, and the mitotic spindle consisting of the microtubules and other proteins extends across the cells between the centrioles. Okay, centrioles. What are the centrioles? These yellow structures that you see here are the centrioles. Okay, so spindles are these structures. These thread-like structures are your spindle fibers. So what I'm trying to tell you here is that the spindle fibers extend between the centrioles to either ends of the cell. Okay, what are the spindle fibers made of? They are just thread-like structures that are made of microtubules and other proteins. And what is their function? They just help in the movement of chromosomes during cell division. All right, just understand that much for now. We have the centrioles, which are these yellow structures here. And we have the spindle fibers, which are these thread-like structures that assist in the movement of chromosomes, right? So that's exactly what happens in prophase. Now, one more thing to note here is that prophase, the name prophase has the prefix pro, indicating that it is the first stage or the first step of mitosis. So keep that in mind. Prophase is the first phase or the first stage of mitosis, right? So I hope that's very clear. Let's go on to the next one, which is metaphase. Now, this is what a cell that reaches metaphase would look like. Now, again, what is special about the name metaphase? Okay, the prefix meta indicates middle. Middle of what is what you may wonder. That's exactly what is the key point in metaphase. What happens is that the chromosomes align themselves at the equator or the middle of the cell. As you can see here, the chromosomes are all arranged at the equator of the cell, right? All of these. The spindle fibers have basically helped them move from where they were inside the nucleus to the center of the or the equator of the cell, as you can see, which is why we call it metaphase. And the nucleus becomes less prominent, or you just cannot see the nucleus at all. In prophase, if you remember, we saw that the nuclear membrane, remember, had started becoming less prominent. By the time the cell reaches metaphase, you don't see the nuclear membrane at all. Okay, so keep that in mind. The chromosomes in metaphase are arranged along the equatorial line of the cells, arrange themselves in the middle of the cell. This is what the actual view is like, and this is what the drawn image would look like. Now, coming to the next phase, which is anaphase. Here again, the prefix ana indicates that it is opposite. Okay, now if you closely observe this image, you would see that the chromosomes that were lined up at the equatorial plate during metaphase have now separated out the sister chromatids. Remember, now for those of you who are not able to recall what we discussed in the previous session about chromatids and chromosomes, let me just quickly tell you what chromatids are. So this is what a replicated chromosome would look like, right? So this structure here in the middle is the centromere. This entire thing is one chromosome. These similar threads that are held together by the centromere are called sister chromatids. Sister chromatids. Right? Keep this in mind. This is a very, very important concept, because without understanding what sister chromatids are, what chromosomes are, what a centromere is, you will not be able to understand what's happening in mitosis, right? So now we are already in the third stage of mitosis, which is anaphase, wherein the sister chromatids from metaphase are separated out and the spindle fibers shorten and they pull them towards opposite poles. Okay, you see that, right? Sister chromatids are individually pulled apart towards opposite ends of the cell. That opposite is what is indicated by the name, by the prefix ana in the name anaphase, right? So we've spoken about prophase, metaphase and anaphase. Now what comes next? We have the last stage of mitosis, which is telophase. Again, here there is a hint in the name. Telo indicates that it is last. Okay, telo, the prefix telo indicates that it's last or towards the end. So here again what happens is, as you can see, the sister chromatids are completely moved towards the opposite poles of the cell, and more interestingly, you can see that the nuclear membrane has begun to reappear. And is it one nuclear membrane as you saw in prophase? No. You see that there are two nuclear membranes. This is one and this is another one, right? There are two nuclear membranes separately surrounding each bunch of sister chromatids, because this is basically aiming to separate it out into two new daughter cells in the next stage. All right? Keep this in mind, children. Telophase is the stage at which the sister chromatids have completely moved towards the opposite poles of the cell and the nuclear membrane has begun to reappear in order to form two new daughter cells. Okay, but you see that it's still not completely separated out, right? There is some connection in between. The cytoplasm is still common between the two. It's only the chromosomes, the sister chromatids, that have been separated out, and it's aiming at forming a new nucleus for each of it. This is what happens in the stage that is telophase. All right? Now, what happens next? We have seen prophase, metaphase, anaphase and telophase. Now coming back to our cell cycle, there is interphase, and we spoke about karyokinesis. Karyokinesis is exactly what mitosis is. The four stages that we discussed just now in the last few minutes fall in this particular portion. Just in this particular portion, prophase, metaphase, anaphase and telophase happen here. So what is next? As you can see, cytokinesis is what the next step is, and ultimately that would lead to the formation of two daughter cells. So let's look at that now. This is what cytokinesis will be like. A furrow completely deepens and a wall is completely formed between the two sets of newly duplicated chromosomes, or the sister chromatids, and the cytoplasm is completely divided so that you have two new daughter cells as the end product of cytokinesis, right? So I have written here final separation by splitting of cytoplasm. Remember what mitosis was also called? It's called karyokinesis because karyon is nucleus. Separation of nucleus is what basically happens in mitosis, and cytokinesis separates out the cytoplasm. And mitosis and cytokinesis together form the mitotic phase or the M phase of the cell cycle. All right. So this is just again to summarize whatever we have done today. We started saying that mitosis is important because it helps in growth, correct? It helps in growth, repair, and different kinds of growth in the body, right? We gave examples of your nails growing, your hair growing, growth, physical growth of all of us from babies to being what we are today, and repair of wounds, any other repair of worn out tissues, all of that, right? Mitosis is the type of cell division that helps in all of these processes. And it happens in different stages, starting with prophase. Prophase is the first stage wherein the chromosomes condense, the nuclear membrane breaks down, and the mitotic spindle begins to form. Those red thread-like structures are what we call the mitotic spindle, right? The second stage is metaphase. Pro is first, meta is middle. And what happens in the middle? Middle, or the equator of the cell, is where the chromosomes are arranged in metaphase. In the third stage, which is anaphase, Ana stands for opposite, the sister chromatids are pulled apart by the spindle fibers and they move towards opposite poles of the cell. And fourth stage is the telophase, telo for last or the end, wherein the mitotic spindle breaks down, the nuclear membrane reforms, and the chromosomes become less condensed. You see that they are already formed into two separate sets of chromatids. And finally, at the end of mitosis, cytokinesis happens, wherein the final splitting of cytoplasm into two occurs and the cell pinches into two new daughter cells. Okay, this is a very, very important concept. I think it does have scope for picture-based questions also, like for example, you may be asked to identify the particular stage. You may be given one of these stages, an image of one of these stages, and asked to identify which stage of mitosis that is. Okay, and if not, you should also be able to draw this in your own way and understand this very clearly in your mind. Have this as a base, children. Again, I'm stressing it is an absolutely important concept, especially for your board exams. Plus, if you are planning to take up biology after your 10th, this is a basic, absolute basic concept that will help you understand a lot of much bigger concepts that will come up in your higher grades. Okay. Now, this is just again quickly coming back to the larger picture of the cell cycle. I'm sure you know this by now. We have the interphase and we have the mitotic phase. The interphase G1, S and G2, which is preparation for mitosis, and the mitotic phase wherein we have prophase, metaphase, anaphase, telophase, followed by cytokinesis. And what do we get at the end of cytokinesis? Two new daughter cells, and individually they would enter into their respective G1 phases, and this would just continue, right? I hope you have this clear in your head now. Now I think we are in a position to define mitosis as a process. Now that we have understood what happens here, we can certainly define this process. We can define mitosis as a type of cell division that results in two daughter cells, each having the same number and kind of chromosomes as the parent nucleus. Thus, mitosis is also called equational division. Now, what exactly do I mean by the same number and kind of chromosomes? If you recall, in the first stage, we had a diploid cell. We are talking about a diploid parent cell. Say, for example, if it's the case of a human being, a typical human cell would contain 46 chromosomes. Okay, the diploid number of human beings is 46 or 23 pairs. The parent cell of one cycle of mitosis is a diploid cell. It goes through prophase, metaphase, anaphase, telophase, and cytokinesis. And at the end of cytokinesis, this would have divided into two new diploid cells. So you see that the number is very clearly, the number of chromosomes very clearly is the same as the parent cell. Even the type of chromosomes would be an exact replica of the parent cell. Correct? This is why we call mitosis equational division. And this is a very, very important thing We have already discussed the significance of it, but let's just put it in points so that for your exam, just in case it's asked, you can easily recall the significance of mitosis. Okay? Now, it's important in the growth of tissues and hence of an organism, as we have seen. Remember that very first picture that I showed you, from a baby to an adult man, the stages that one goes through, right? It is growth of tissues and as a result, growth of the organism as a whole. Secondly, it maintains the same number of chromosomes as that of the parents. So, for instance, every human being has to have the typical number of 46 chromosomes in every diploid cell. It is only if mitosis happens that this number can be maintained with every generation. Equational division is the method by which this can be ensured, right? The same number of chromosomes in the parents and offspring. Thirdly, helps in the repair of worn out cells. Again, we have seen this. And another point that we have not discussed is it's significant in cancer research, because what exactly happens in cancer? It's basically uncontrolled cell division, right? And why do cells divide uncontrollably? Basically because some checkpoints that has to put an end to the cell division in that particular part is not functioning due to various reasons, which a lot of research is happening on that. But basically, cancer is uncontrolled cell division, correct? So, uncontrolled cell division basically means it is uncontrolled mitosis. Because of this, mitosis, the study or a deeper research in mitosis has been a very, very significant aspect in all kinds of cancer research. So these 4 points, keep it in mind. It can be asked in your exam. Now, this is your homework question for today. Identify the following stage of mitosis. Let's see who is answering first. Well, this is a very objective answer, so I don't think there is a question of getting the best answer for this particular question. Let's see who gives us the quickest answer. Okay, so I hope you have understood all the stages of mitosis, children. Do revise this, understand this concept very clearly so that we are also in a much easier position to understand the next concept, which will be meiosis. Okay, so do not forget to download Vedantu's learning app. If you have not done it already, do join our Telegram group so that you can chat with our teachers directly regarding any of your academic doubts. And we do have a lot of other amazing features out there. And most importantly, if you found this video useful, if you found this session useful, do not forget to hit the like button. Do share it with your friends and whoever you think might benefit from this. And if you have not subscribed to our channel, do hit the subscribe button. Vedantu 9th and 10th, that's our channel, because we will be coming up with a lot more interesting sessions, a lot more live and premiere, of course, a mix of both based on your ICSE topics in the days to come. So make sure you stay tuned to our channel and do not miss any of our sessions. Just in case you have any questions regarding what I have explained today, you can either send me an email on ambika.gopalakrishnan@vedantu.com, or please feel free to put your comments in the comment section right below this video. Right? So I'll see you again in another interesting session. Until then, happy studying and bye-bye.